Hello everyone, my name is Srikar. I am a graduate student from Kansas State University and also a member of Concordant Lab, which mainly focuses on semantic web technologies. Today I am going to present our research entitled Generating Reliable Adverse Event Profiles for Health through Automated Integrated Data, which is also known as GraphAid. So it is basically a semi-automated knowledge graph building approach, which we are implementing on adverse events data. We all know what is an adverse event. An adverse event is an unintended, unfavorable, or harmful outcome that occurs when a patient receives medical care. They can be caused by medication errors, diagnostic errors, or different combination of drugs and vaccines. Adverse events in drugs may be related to vaccines or vice versa, but there is no effort in trying to put these two data together. The data for the adverse events is increasing day every day, and there is also frequent changes in the content of the data. Connecting this drug and vaccine data for human health is critical. So the main focus of this paper is on adverse events caused by using the combination of drugs and vaccines and find out if they are related in any format and try to connect them both using a semantic web approach that is semi-automated. For that, we want to use knowledge graphs because they are versatile, they are easier to integrate as they can extract entities and context from new data and automatically update the knowledge base in response to data changes. For machine learning, knowledge graphs can improve machine learning and AI algorithms by providing structured and context-aware data, which can lead to more accurate and nuanced results. For user experience, knowledge graphs can provide a wide range of information on a concept, which can reduce the number of clicks and time required to find the matching content. To create a knowledge graph, it is really important to follow the systematic methodologies because there is constant change in data and there will be new data to integrate in your knowledge graph. If we do not follow a systematic methodology, then we may need to repeat the same work or even start from the beginning to add new data. In order to create and update the knowledge graph in a systematic and modular way, we use the knowledge acquisition and representation methodology, also known as KNARM. KNARM is a 9-step methodology that allows domain experts and knowledge engineers to build useful, consistent ontologies formalizing domain data and knowledge. Some of the main steps of KNARM are sublanguage analysis, which identifies the key concepts and terms used in the domain. The data which we have taken are FDA Adverse Event Reporting System, also known as FAERS, and Vaccine Adverse Event Reporting System, known as VAERS. These two datasets are taken from the US Food and Drug Administration. These datasets include adverse events caused by combination of drugs and vaccines, respectively. After collecting the data, we have done an unstructured interview, which includes knowledge from domain experts. So we worked with our FDA collaborators to identify different parts of the data. The data from the FDA is in XML format and it is structured yet in nested format. Each element contains an array of elements. As shown in the figure, each safety report element consists of several elements like patient elements, and each patient has other elements like reactions reported by patients and the drugs which they took. After that, we have done sublanguage recycling, which uses existing ontologies or vocabularies related to the domain. There is an ontology for adverse event, which is the ontology of adverse events. It is a biomedical ontology in the domain of adverse events. The adverse event ontology aims to standardize adverse event annotation, integrate various adverse event data, and support computer-assisted reasoning. The adverse event ontology is a community-based ontology. And we also looked into vaccine ontology, which is also a community-based biomedical ontology in the domain of vaccine and vaccination. Vaccine ontology aims to standardize vaccine types and annotations, integrate various vaccine data, and support computer-assisted reasoning. The vaccine ontology supports basic vaccine R&D and clinical vaccine usage. The vaccine ontology is being developed as a community-based ontology with support and collaborations from the vaccine and bioontology communities. And we are still in the process of identifying how the vocabulary in the FDA datasets are maybe matching with those vocabularies in the ontologies and also reusing them. So there are no mappings or immediate usage for our FDA dataset right now. So these mappings will be completed as one of the next step of this work. Then we also done modeling based on the structure of the FDI dataset. We did our initial modeling. Our initial model includes patient node and it is connected to the drug node and outcome or reaction node. As shown in the FDA dataset previously, it is similar to the patient elements, contains drug elements and reaction elements. Each of these nodes have their own properties and child nodes connected to them. Using this, we are still collaborating with the FDA to kind of follow through with this. But as we are working on the steps, we are working on the database formation, which is the backbone for the representation of the knowledge. And it is what this presentation is focusing on. So for our database, we have chosen Neo4j database. It is a graph database and it can handle intensive data relationship. So how did we create this database? So in the initial step, we have taken the data sets from the FDA. As we have discussed, the data is in nested structure, and there is also data inconsistency, such as some patient reports does not contain all the necessary information. Since we are trying to load this data into Neo4j, which does not support XML, we decided to convert the data into JSON structure to not flatten out the structure of the data. So we also removed inconsistencies. All these steps were done by implementing a Python script. The resulted file from this Python script is a JSON file. So then we have implemented a Cypher query on this JSON file, which uses Apac library to handle JSON files and use functions like unwind and merge to load the data into the Neo4j. After loading the data into Neo4j, we are able to query different types of research questions within our provided data, such as list of patients who reported myopericarditis and type 2 diabetes. As you can see in the figure on the left, the red color nodes are reaction nodes and blue nodes are the patient nodes who have reported the adverse events, and the yellow nodes below are the drug nodes which the patient took. And we can also query two patients who took the common drugs, etc. We did the same thing for the vaccine data, such that we have extracted the weights data and imported that into the Neo4j database. And we compared it with the drugs data. They both are structured in the similar fashion, where a patient node is related to vaccine or drug node and related to the reaction or outcome node. There is a potential in connecting these two data together. As we all know that to convert the data to a more meaningful format that allows inference of new knowledge, which is not explicit in the flat representation in the database, it can be achieved by using semantic web technologies, mainly an ontology. So after placing the data dissected based on the metadata as well as the metadata into the database, we tried to convert our data to the semantic web approach. So building a knowledge graph is particularly relevant for representing complex knowledge involving hierarchies of concepts and many specific relationships among concepts and their data properties. However, to create an ontology is a tedious task, and the KNARM methodology highlights some challenges. There is no smooth integration of Neo4j database with OWL. There is also some complexity of DL syntax and learning curve for the users. We have taken an approach of using Python and its RDF library for knowledge graph development. The key features of this is to automatically generate classes and their axioms from the Neo4j data and provides smoother integration process. For that, we have first made connection with the Neo4j database and fetched the data in sessions. We have applied RDF library functions to initialize the graph and namespaces. We have defined the classes, object properties, data properties, and implemented the logic to automatically set the subclasses for their respective classes and object and data properties to those classes. We can download the resulted out file from our script and run it on the protege as shown in the figures. Uh, as shown in the left figure, we have classes for adverse event, drug, patient, and safety report. All these subclasses, which we can see on the right side, are automatically populated from the Neo4j database using our script. Uh, as you can see, the usage section, the object and data properties are also populated as well, which is automatic. Right now, the script runs explicitly for the FDA datasets only, and we are still in the process of validating from the domain experts. So our main focus of this paper is to connect FAERS and VDOS data set using semantic methods and to find what combination of drugs or vaccines causing adverse event. In the process of doing so, we also incorporated a semi-automated way of creating an ontology. And thank you so much for giving me this opportunity. And I would also like to thank our collaborators from the FDA. And if you have any queries about how our scripts work and about any questions, you can contact me on this email ID. Thank you. Any questions?
Great. Yeah, good talk. So any questions?
Yeah, I have one. So, maybe again I missed. Did you use the vaccine ontology and the OEA, the ontology for adverse OAE, to map to your data? And is there any difficulties?
We are still working on the modeling and final ontology process, so we are still not. I mean, we did not make sure that we can use these vocabularies from these ontologies. So we are just in the initial stages of modeling.
Oh, so you mean you are not so sure that you will use those ontologies? Did I hear it right? You said you are not so sure. You are just evaluating. Oh, okay.
We are in the process of mapping.
Yeah. Okay. And also you have all those drugs, right? You said there's data inconsistency. What the data inconsistencies that you found?
I mean, as you can see in the data set, some patients does not have all the drugs which he has taken or she has taken, and it may lead to inconsistencies like validating through machine learning. So we try to remove those inconsistencies.
Yeah, and another thing is about the FAERS, all those VAERS reporting system. They are passive reporting system. The data is just incomplete, inconsistent. That is their nature. So I don't know if you remove the inconsistent data. I don't know. Yeah. So there has to be something.
Yeah, we have removed the inconsistent data using implementing a Python script. So
yeah, I don't know if you remove those inconsistent data will have an impact for the future. Yeah, future. What I meant is the data is it's incomplete in nature. So, and therefore there are specific statistical analysis to see the adverse events to the causal relations. And I know the FDA, your collaborators, they know all this. So did you consider? Yeah, I don't know. I just think you probably need to think about that. Yeah. I'm just a little bit concerned if you just remove all the inconsistency, then the data may not really reflect anything, and then you may not get the right answer that you want to ask. That is, yeah.
I mean, the inconsistency, as I talked before, so the inconsistency is based upon the incomplete data in the patient elements. So we have implemented a script so which automatically detects the inconsistency in the patient elements and it removes that inconsistent data. So that's what we try to do. So
yeah, what I meant is that because the data itself has this nature, and there are specific statistics methodology to design for analysis of adverse events that related to a certain drug or a certain vaccine. And I'm just concerned that if you remove all those, what you think is inconsistent data, and it may impact the future result. That is, yeah.
I mean, some patient notes have they have taken these drugs, but the reaction elements are incomplete. Like it's like just blank. So it may not be so much useful while analyzing that data.
Okay, thank you.
Okay, yeah, so that's great. Yeah, so yeah, I have a quick question. So I'm wondering what's the additional benefit of kind of combining the FAERS and the VAERS. So one is drug, right? One is vaccine. So in nature, they are often different. So I'm wondering, like because often there can be some interaction between drugs and vaccines. Maybe some people do vaccine and having the drug together, but somehow by combining data, you may find something interesting. Yeah, I'm just wondering, yeah. It's interesting talk.
Thank you.
Okay, so I think, yeah, probably it's about time now. So any other question? Quick questions?